So first of all are that what are these resistant mechanisms and how do we counteract these resistant mechanisms? And what is the Achilles heels here? So when cancer cells become resistant to some mechanisms, they develop an alternative mechanism, right? And that is why they can survive those conditions. But when they develop that alternate mechanism, there could be something vulnerable about that alternate mechanism. And if you can identify that kind of vulnerability, you can attack that weak part and you can kill that. So the normal cells don't have that new path, but this new path may also have its own weakness. And if you can identify that weakness, then you can attack that weakness. And that is what is meant by Achilles heel here. So there are two kinds of resistance in cancer. There is intrinsic resistance, and then there is acquired resistance. So suppose you develop a drug, right? And you give it to two different patients. One patient responds to that drug, the other patient does not respond to that drug. So one of the reasons is that the other patient who is not responding to that drug, he is resistant to that drug. So there is something internally present in that patient which makes him or her resistant to that drug. So that is intrinsic. The tumor itself is resistant to start with, even before the treatment. Then there is acquired resistance. You keep on giving the drug, and like I was saying for Gleevec, those patients then develop a resistance mechanism. Okay, so that is acquired resistance. So for example, when you have intrinsic resistance, one of the reasons why this is always present there, why they are not responding in the first place, because they don't even have that target. Suppose you are giving against BCR-ABL, and if it doesn't have BCR-ABL, it's not going to respond to Gleevec. Or there is some mutation in the BCR-ABL in this patient so that it does not bind Gleevec. So again, so it is low affinity. It does not even binding that. Or they have some mechanism so that the drug is not even getting inside the cell. So this cell is not letting the drug come to that. Or there is something above the cell which is blocking access of that medicine or that drug to come inside the cell. Or there are many receptors here which throw the drug out. So what happens when you give a drug, and this is the body's defense mechanism also sometimes, that there are these receptors, efflux receptors. So you give something, they just throw it out. You give it, and so initially let's say there are two of these things, right, in a cell. But when you are exposing them to the cells of now, many of these things will come and they will just throw it outside the body. So that is called efflux mechanism. They throw the drug out. So this is a very common thing. When you give drugs to the patient, these guys are going to be upregulated and they will throw the drug out of the body, out of the cell. So that is intrinsic resistance. And this efflux mechanism could be due to acquired also. It wasn't there initially, but when they got exposed to the drug for a long time, now they start throwing the drug out of the cell. Okay, so that is why efflux mechanism is also here. Okay, acquired is that initially Gleevec was working beautifully, BCR-ABL, but then these patients, after just 3 months of Gleevec, they developed resistance because now BCR-ABL mutated, now Gleevec doesn't bind. Okay, so or they have developed new mechanism that they don't even depend on the first target. So they have alternate. So suppose I have this mechanism, you block this mechanism, I develop this mechanism. So suppose you are buying your chemical from Sigma. Sigma stops selling it, you buy through someone else, right? So another pathway, okay? Or the target itself is modified. So there are many ways to acquire resistance. So this is intrinsic resistance, intrinsic mutation. It was there from the start. And so initially when you gave the drug, most of the cells die, but these guys which were resistant because they already had that mutation, these guys will make a new tumor. Now you can keep on adding the drug, nothing will happen. Here, these guys were pretty naive. You gave it, some are survivors, right? We know some are survivors. So they change themselves to survive the conditions, right? And now they make the treatment, they are resistant, okay? So key drivers, so the problem of resistance, so genetic mutations occurs, the protein modifies itself, introduce new mutations so the drug cannot bind there. Epigenetics rechanges, right? They will change the DNA methylation pattern, histone acetylation or deacetylation pattern, and they will activate new genes and things like that. So they will rewire the system. Okay, tumor microenvironment support. So once again, let me remind you about the tumor microenvironment, and I'm going to discuss a lot about this thing. This is a relatively new field. So this is cancer tumor. This is the tumor, right? So this tumor alone is not responsible for cancer, right? This is, you can think of tumor as a very, very, very super smart guy. So this is the tumor and this is going against the body, right? So it is so smart. If it encounters some drug, it will mutate to protect itself. It will steal from other guys to survive because it is growing so fast. So there is a lot of stealing going on from normal cells. It fools the immune system. It acts and it tells the immune system, I am just part of you, don't kill me. Okay, and then it will recruit all the things to shut down the immune system. So this is the tumor, but a lot of it is going outside the tumor. All the environment outside is supportive to the tumor, and it will send off all the bad signals from outside. It will protect itself from many different things. So that all forms the tumor microenvironment. So immune system helps us, right? You know that. So there are many cells which are considered anti-tumorigenic. So if they come, they will kill the tumor. So tumor cells will secrete something and make anti-tumorigenic cells pro-tumorigenic. Now they have changed to help them. So not only it is like you take the enemy and you make it your friend. So it has actually come to kill the tumor. Now it is helping the tumor to grow even bigger. So that is all because of change in the tumor microenvironment. One of the things they do, they secrete a lot of acid. So the whole environment is acidic. And in this acidic environment, nothing can come close to that, which can kill the tumor. Okay, and then if you make too much acidity, things will break apart, tumor can spread around. Okay, so that is all comes under tumor microenvironment. And phenotypic plasticity, that suppose somebody is considered a culprit in one country and that person changes the name and completely hides itself when he's or she's living in the society but nobody knows. So that is phenotypic plasticity, just completely change your appearance. And then the drugs cannot recognize you because you have changed. You don't depend on the protein that the drug is against. That is phenotypic plasticity, okay? So let's discuss a little bit of many different mechanisms of resistance. So genetic mutation, you have changed the target itself. Drug will no longer bind there. Gene amplification, now you make a lot more protein than before. So initially you were making this much protein, the drug was effective. Now you're making so much protein, the drug cannot even compete with that. Okay, you can bypass that. Suppose the drug was, suppose this cancer was due to something. So you give so like prostate cancer depends on androgen receptor signaling. If you give androgen receptor signaling inhibitors, after some time they don't even care about androgen receptor at all. They completely forget about androgen receptor. They don't express androgen receptor, they don't depend on androgen, they don't care about it. There is no androgen related signaling there. Now they overexpress MYC protein, and MYC is the one which is leading to cancer. You can keep on adding androgen inhibitors, who cares? They don't need that anymore. So they just bypass the whole signaling pathway. This is the bad way. They overexpress efflux-prem. So you add drug, they throw it out. You add drug, they throw it out. Okay? This is another thing. So whenever you think of a tumor, tumor cells are not all the same cells. there is huge heterogeneity. That means that there are many different things which are wrong in this tumor. So suppose you're giving drug against that. Okay, it will kill this part of the thing because this part of the thing depended on that target. But there are many different targets in different parts of the tumor. So tumor is so heterogeneous. Even if you kill one part of it, it's fine. The rest of it will still grow and keep dividing, and the tumor will keep on growing because the tumor is really heterogeneous, right? And then, so let's do radiation therapy. We give so much radiation, will damage the DNA, tumor should disappear. Now they will upregulate DNA repair enzymes, tumor cells. You give them damage, they repair it. And you know what? If they inefficiently repair it, no problem. They will make more mutations, adapt, move on, right? So, and then they will do epigenetic rewiring. Again, oncogenes are overexpressed, tumor suppresses down. So global hypermeasurement. So they can do a lot of things. They will inactivate tumor suppressor. They will activate genomic thing. So they can do all this epigenetic rewiring to support cancer. Okay, and keep in mind, you know the survival of the fittest. So even if some die, who cares? Those who die, they die. You are not meant to be part of the cancer thing. I'm just telling you cancer philosophy. Those who survive are the ones which will make more. So whoever survive, even if there is one single cell, plenty, that one single cell will form the whole tumor, that one single cell will metastasize, right? So if they are dying, nobody cares about dying cells, because that's why I was saying slowing is sometimes the best option. But if you can kill it, that is absolutely the best option. But cancer can be so devastating that slowing itself is considered really a big win, right? Especially when some cancer comes unannounced and the person doesn't know anything and they think, they have 20, 30, 40 years to live, and then cancer shows up. So then even if you can extend life by 6 months, it's a win, as long as the quality of life has not deteriorated too much. Okay, so now let's look at the tumor environment. So look at this part here, okay? So what this is telling you, that there are many different kinds of cells which are present in tumor microenvironment. This is a picture I generated using AI. So AI always messes up the picture, but it was still beautiful enough for me to put it here. Okay, so here you can see look at all of these fibrous structures. This is not ideal, but basically a tumor is surrounded by hundreds of different kinds of cells which support the tumor growth. Okay, and those are extremely important for protecting the tumor. So if you can actually attack the tumor microenvironment itself, you can reduce cancer. Okay, so now let's talk about strategies to counteract these resistance. So, so first you figure out exactly what is the cause of cancer. Okay, this is very important to figure out what is the cause of cancer before you make your plan of attack. So for example, like I was telling you yesterday, in pancreatic cancer is the most lethal cancer. So pancreatic cancer does not occur in many patients. So if you take, I don't remember the number, in terms of cases, pancreatic cancer is maybe the 10th it is 10th in number, as I recall. So not many patients get pancreatic cancer, but if they get pancreatic cancer, it is the leading number one cause for cancer-related death. So most of the patients who get pancreatic cancer do not survive. Less than 5% of people have a survival rate for more than 5 years. Breast cancer, it could be 80% people survive, 85% people survive. For pancreatic cancer, it's less than 5%. It's the most lethal cancer. So this is the tumor, and this big is the microenvironment, this big. So they are surrounded by a lot of other cells which protect the tumor. So what people thought, that they poke hole in these whole fibrous mess, and they can get the drug out to the pancreatic cancer cell and it will die. and you can treat it. But the problem is that this is what I meant by you have to have a plan of action before you attack cancer. So if you poke hole, right, if you break the fibrous thing to get the drug inside, what is the side effect? Now you are making it a channel for pancreatic cancer cells to escape and form tumor elsewhere. So it's not a good idea to break also that, because so far it is more contained because of this whole fibrous thing. But breaking open it so that it dies can also lead to metastasis. So you have to have a very solid plan to attack cancer. Okay, so that's what it is saying, that you have to really figure out using genomics, proteomics, metabolomics, all the tools that we have to find the real cause for that cancer, and then you can decide exactly what kind of therapy you are going to use. Okay, I know of someone who was diagnosed of very aggressive cancer recently, and by the time it was diagnosed, it had already spread to other organs. So it was imperative that before going and really figuring it out, which would take at least a month or two, and the cancer was spreading so fast that patient didn't have 1 month or two just to figure out the thing. So what they did before figuring out exactly what went on, they started immunotherapy because they wanted to contain the disease. And now that person is responding to immunotherapy, right? And the disease has stabilized. And it is also regressing a little bit. So sometimes you have to make a decision to make it stabilized. Don't let it spread. But at the same time, you should also make a priority to figuring out exactly what kind of precision medicine is needed. Okay. So cancer cells undergo a lot of things to have all these bypass mechanisms to avoid that. So how can we identify the weakness of cancer? So in ideal situation, normal cells should not be affected by the disease. Cancer cells should be affected by the medicine, right? So that is the whole point. So I have already told you about the resistance mechanism, but I will tell you how do you overcome drug resistance. So we can sequence the DNA to find out what kind of mutation it is. We can use CRISPR-Cas9 to change the DNA. These things are not easy, but these things are being used. Okay, so we can change the DNA. We can do high throughput screening to find the drugs. Somebody, you were asking me, right? Whether we should do molecular modeling first. So the way pharmaceutical industries, they identify drugs towards target, they have so many. See, in our academic labs, we make 10 molecules or 20 molecules or 50 molecules. That's a huge amount of work to make 50 molecules, 50 different molecules. But industry, they already have millions of compounds in their library. So they do high throughput drug screening because they already have compounds in hands. And then they see, okay, these are the hits. And then they start from those hits. And then they modify the hit to make it even more potent compounds. So you can do high throughput screening to identify. You can do combination in high throughput screening and see what two drugs in combination can be more effective. So this can give you results in weeks, right? Combination. But then obviously you have to test it and everything. Okay. Now this is something I have never talked about. This is biomarkers. So we have a whole lecture. Probably tomorrow I'm going to tell you about biomarkers in details. But biomarkers are basically will tell you what kind of therapy will be good. So for example, remember I told you about HER2. So HER2 is a biomarker for that cancer. If that patient is expressing HER2, you know we have to give Herceptin as a disease because Herceptin is an antibody which will bind HER2. So your biomarker is telling you what therapy you should use. Sometimes the biomarker can tell you whether the drug is going to be effective, how much effective, whether you should change the dose. All of these things come under biomarkers because there are many, many different kinds of biomarkers. So biomarkers really help in precision therapy. Okay, So, and then we can combine it with proteomics, genomics, immunotherapy for cancer. Okay, so these are few different ways you can tackle cancer. So instead of giving one drug, you give combination of drugs. So do you know what is synergistic and additive effects? Some of you know. Do you know? All of you? Probably not. So suppose you give A drug and B drug. A is working, B is also working. But if you combine them together, so this can be additive. If this was 5%, this was 5%, you combine them, now it becomes 10%. So 5 plus 5, 10. Additive effect, right? Or when you combine this and this is 5% alone, this is 5% alone, but when you combine them, this becomes 50%, synergistic. So this is not additive, it is synergistic. So it is much more than the additive effect, that is synergistic. This is ideal situation to combine them, and there are many cases where you use combination therapy. Or sometimes this is 5%, you combine it with this is 5%, now it is 2%. That means these two cannot work in combination. They are basically killing each other, right? You don't want that. So combination therapy, you have to figure out what combination. Sometimes it could be a combination of 3 drugs. I know cancer patients are usually given 2 kind of therapies together, angiogenesis inhibitors and immunotherapy, and that is very effective, okay, because you are stopping the progression by inhibiting angiogenesis and you are also treating the cancer. So 2-prong attack is very effective, okay. So that is combination therapy. Immunotherapy is very effective. This is a game changer in cancer field, immunotherapy. And I briefly told you about PD-L1 therapy yesterday and CTL4. We'll come back to that more when I tell you about immunotherapy. Okay, tumor vaccine. So here we were discussing vaccine during the break, right? So if you take tumor-specific protein and you make a vaccine against the tumor-specific thing, normal cells will not be affected, tumor cells will die. Okay, then gene therapy. If tumor suppressors are defective, you introduce a gene to express tumor suppressors. That would be a gene therapy. If you have an oncogene, you can send an shRNA to to degrade the oncogene, right? To degrade the mRNA of the oncogene, you basically are removing that. Either you can use an inhibitor to shut down the protein, or you can just get rid of the protein altogether. So somewhere, I don't know which day, because we are going at a rapid speed, but I'll tell you about PROTACs. And in PROTACs, you make the protein disappear, okay? But in this one, it is showing you that you can degrade the RNA. Antibody drug conjugate. So antibodies, this is very powerful method, antibody drug conjugate. So what you can do, what is the feature of antibody? So suppose this is protein A and you make an antibody, and you can have millions of other protein, but this antibody will only bind this protein A. This is so specific. Okay, so suppose you make an antibody towards a protein, but you also hide the drug in the antibody. So now the antibody will go to the tumor, bind, and the drug will be delivered here, and this tumor will die. But you don't know what drug to deliver. You attach radiation therapy, it will directly go to the tumor. Or you can attach an imaging agent and the tumor will light up. So even if you have a single cell, that single cell will show in the imaging because you have light up the tumor. So that is antibody conjugated drug, ADC. Right? So people attach all sorts of drug and deliver it directly to the tumor. Okay, then targeted therapy like HER2 therapy I just showed you, right? Receptors overexpressed, you target those ones. So this is targeted. BCR-ABL, targeted therapy. We are not giving chemotherapy, we are just giving the drug. BCR-ABL is not present in normal cells, so you are actually only giving it to cancer cell. That is targeted therapy. Okay, you can introduce different kind of cell death to tumors. Okay, This is very important. CSC and EMT. CSC is cancer stem cells and EMT is epithelial to mesenchymal transition. So think of normal cells like bricks, right? So these normal cells, they look like, these are cancer cells, but they look like this, right? They're pretty, wide and everything, more organized. So they cannot go through the blood vessels easily because these cells are not very mobile. So they undergo a process which is called epithelial to mesenchymal transition. So these cells, they become more mesenchymal. So if this is the normal cells, I also have a slide to show you this thing. They will become like this kind of cells. Look at the shape here, really different. So this is epithelial morphology and this is mesenchymal. Okay, so what is the advantage of this transition, epithelial to mesenchymal? Mesenchymal, they are very narrow, so they can go through this hole and they can metastasize, they can get into the blood vessel. Plus, they are very motile. Epithelial are not very motile. Mesenchymal is very motile, so it can squeeze in through the space, go out and metastasize in other organs. And once they reach some other organ, they will become epithelial again and expand the colony to form a new tumor. So that is EMT. When EMT occurs, cancer is considered really bad. And cancer stem cell, so remember, stem cell can differentiate to any cells. So when cancer, so what are cancer stem cells? So suppose this is the tumor, right? And you give radiation therapy or any therapy and you kill 99.99999% of tumor cells, but literally one cell survives. Just one cell survives out of the trillions of cancer cells. That one cell is resistant to radiation, resistant to drug, resistant to that, and that one cell is the cancer stem cell, which is just resistance to everything, and that can make a whole new tumor. And those cells are very hard to kill. But now we also know how to kill those cells because we know what exactly goes inside those cells and which proteins make them so resistant. So attack those proteins, even those cells will die. Okay. And lipid nanoparticle is just a way to deliver the cargo to the right location. Okay. So let's talk about different ways. So I'm going to tell you about 5 different things. Inhibitors, so like Gleevec was very effective. Resistance emerged, then they make 2nd generation, 3rd generation, and one of the very common one, now nobody uses Gleevec, they use Dasatinib. Gleevec was called Imatinib, and then Imatinib was converted, same scaffold, same thing, new changes around the same molecule to counteract the resistance. So Dasatinib is the next generation inhibitor, and this is even more effective than Imatinib, Gleevec. Okay, so you can make more and more modifications. So these are called next generation inhibitors. So 1st generation inhibitors you modify, 2nd generation you modify them, 3rd generation. So cancer is modifying, and so we are modifying the molecules accordingly, right? Combination therapies, synthetic lethality, PARP inhibitors, we'll tell about that. You will love the synthetic lethality section, and then we'll talk about tumor microenvironment and epigenetic modifiers. So next generation is very simple. You give the drug, resistance emerges, and then you modify the compound. Okay, so that is, so what is the characteristic of better inhibitors? They have better selectivity, right? So when you guys are making inhibitors towards it, right, it is very easy to identify inhibitors. The main thing is that it is only hit your target and no other target. So selectivity is more important because oftentimes if it is non-selective, it is also hitting normal cells because it is binding other targets, right? If there is resistance, that the inhibitor should be modified so that it can overcome resistance. Improved safety, it should not be toxic, right? If it is selective, it is going to be less toxic. And allosteric inhibitions. So what happens, especially in the kinase field, all kinases have ATP binding pocket. So many kinases, the pocket looks very similar to each other because they all bind ATP. But they all have allosteric pockets, like pockets outside of the catalytic domain. So if you make something which will bind allosteric site, those are very unique to kinases. So those inhibitors tend to be more selective. So sometimes instead of targeting the active site, you can target other pockets. Do you guys know what is allosteric inhibition versus competitive inhibition? Everyone? You do not so clear about it, right? You guys appear apologetic, but I'm not teaching chemistry, because then these guys will be asking me the same question. So ask me anytime, okay? All the time, if it is not clear. So what happens in allosteric, that this is the catalytic site, right? And imagine this whole hand is the protein. So what happens is that something is here, there is a pocket here, and if something binds here, this closes, okay? And something binds here, this opens up. But this two part is not part of this, they are very far apart. But binding here or here is controlling whether it is turning on or off, is it right? So that is allosteric. It's not in the active site, it is further away, but this binding is changing the conformation of the enzymatic active site. Is it making sense? So that is allosteric. So you can make inhibitors two ways. You can directly block this, right? That is competitive inhibition, because that's where the substrate bind, you are competing with the substrate. But if you bind here or here, that is allosteric. Very simple words, right? So allosteric sites tend to be different from one protein to the other. So you can achieve more selectivity. That's the point here. Sometimes allosteric sites can be deep buried in the pocket. So they are not directly controlling that, but if you target that pocket within the active site, you can achieve higher selectivity. That's what it means. Okay, simple words. So these next generation inhibitors, they target these specific things, right? So all your classroom, they look the same, but this classroom may have something special there. So you can make use of that special thing. So most of them are rectangular classrooms, right? They have the same shape, but every classroom, even in this building, has something unique about it, right? And that unique, it is still the classroom. When you're sitting here, you're not accessing that pocket, but this active site still has that special thing there. That's allosteric site, right? So that site is not helping all of you sit here, right? So if ATP is coming here, that allosteric site is not really helping ATP, but it has the uniqueness hidden there. That's the allosteric part. Okay? Okay. So this is just showing you, next generation inhibitors, and it overcomes. So this is third generation, fourth generation. So every time cancer mutates, we also change the inhibitor. So we keep on calling them next generation inhibitors. Okay, now let's look at combination therapies. Okay, so in combination therapy, it could be additive, like 1 + 1, 2. So this is 5%, this is 5%, you combine, it becomes 10%. Great, better than 5%. Okay, it could be independent. So that these two drugs then don't work in the same pathway, but if you use them together, it's like immunotherapy. I was telling people are given immunotherapy and they're giving VEGF inhibitors. VEGF means angiogenesis inhibitors. So they are two different things. Immunotherapy is activating the immune system. This is stopping the growth of new blood vessels, totally independent. But both are killing cancer. Okay. And then synergistic, this is best. 5 + 5 together is 50%. That's synergistic. That is very, very good. Okay. And antagonism, once in a while, many times if you don't combine them together, it is very bad for cancer. The drugs themselves kill each other. Okay. So advantages of combination therapy, of course, if you use the right combination, it is more efficacious. Okay, you can kill cancer fast, you can overcome drug resistance. Okay, so now they are trying to develop drugs, which is like you just hit cancer cells with a brick load of things. Actually, brick loads of things. Imagine a truck with bricks, and you come and just you dump all the bricks all together. Like in no time, the whole brick is given there. They are going to collapse with the weight of those bricks. So it's like the whole, and these are actual that kind of thing. So they assemble a lot of things together, right? It's like a huge thing. And then cancer cells express the enzyme, which is only specific to cancer cells, and that just lights the fire here, and the whole thing collapses like this, and the cancer cells just die right away. Okay. So then they don't develop resistance, because they can develop resistance if they have some time to develop resistance. But if you attack them so suddenly out of nowhere, the whole thing dies. Okay, you target multiple population. Remember, tumor is heterogeneous. If you inhibit one thing, the tumor may not regress. So if you're attacking from multiple directions using combination therapy, you can kill the tumor quickly. If you use the right combination of drugs, it may be less toxic. If you use the wrong combination, it will be very toxic. Okay, and this has improved patient outcomes. Okay, what are the challenges? You have to choose the drugs very carefully. The drug should not interact with the drug itself and kill each other. So drug-drug interaction, and the more drugs you use, there is a chance of more toxicity, more complexity. Okay, and like we were talking about vaccine, like you give vaccine for the right cause, but it can also cause its own thing. Okay, I'm a pro-vaccine person, but there is always a possibility that vaccine itself will introduce more complexity in some patients. Okay, and if you use more, it is going to be more costly, right? So that itself could become an issue. And then again, every person is different. Most of the people are benefited from vaccine, but there are always some patients, some percentage of patients who develop long-term complications with even as simple as vaccines. Okay, so type of combination therapy. So these are very common type of combination therapy, that people use chemotherapy and radiation together just to kill the cancer cells. Okay, immunotherapy is often used with angiogenesis inhibitors. Immunotherapy can also be used with chemotherapy. Immunotherapy can also be used, it doesn't show you here, but it is used with many other combinations. So basically to kill the cancer cells. Targeted therapy, sometimes you use two targets at once. Sometimes what happens is that you are inhibiting this target, but you are also inhibiting the target which is below that. So if this escapes that, at least you kill this. So if you kill two in the same pathway, the chances are the whole pathway will shut down. Okay, chemotherapy and immunotherapy are mixed together. Chemotherapy and angiogenesis therapies are mixed together. Radiation and immunotherapies are mixed together. So these are different kinds of combination therapy, but in combination, remember yesterday I also said sometimes before undergoing surgery, people give chemotherapy to reduce the tumor before surgery. Sometimes they do the surgery, then give chemotherapy. If something has escaped, they will die. So they combine different kinds of therapies. So radiation therapy, chemotherapy, immunotherapy, targeted therapy, they are often used in combination. Okay. So rationale for combination therapy, you are attacking cancer from multiple directions. Okay. Now we come to very interesting topic called synthetic lethality. So what exactly is synthetic lethality? So in very simple words, so do you have a class? Oh, you are for this class. Okay. Usually when you teach a class and students come towards the end, I forget, what time did we start?
Yeah, 12:05, so 1 o'clock.
1 o'clock, plenty of time. Okay, so this is a very interesting topic. So our cells are very smart. You shut down one pathway, they will do the same thing using the second pathway. Normally it is a good thing. So here it is showing you that in normal cells there are two ways to reach to this thing, to keep it alive. So if you attack only A, B will predominate, it will take over that thing. It's like, like two people are doing the same job and one person goes on vacation, the other person will take care of that, and the other person goes on vacation, the first person will take care of that person. But if both the people leave, the work will stop. So if you target either A or either B, cells are alive. But if you target both of them, it is lethal, right? So that is very easy to understand, right? And it makes sense, right? Now what happens in cancer cells, that normal cells, so here in this case this is normal cells, gene A and gene B, they are both functional. So if you inhibit only gene A, normal cells will survive. If you hit only gene B, normal cells will survive. Now here is the thing, remember I said Achilles heel for cancer. So what in cancer cells, what happens that, so here it is showing you if you make mutation in gene B or gene A, in both cases they are okay, right? But if you kill both the genes, then they will die. So now let's go to cancer cell. So this is normal cell. So normal cell, if you have gene A or gene B only, so if you hit only gene A, then it will survive. If you hit only gene B, it will survive. But if you hit both of them together, they will die. This is normal cell. Now let's come to cancer cells. So gene A, gene B are required for viability. If you hit gene A or gene B, gene A, it will still be viable. But here in tumor cell, gene B is already dead. Okay, they rely on gene A for cellular viability. So now you make drug against gene A. So normal cells will live because they still have gene B, but tumor cells will die because they don't have gene A to start with. Is it making sense? I'm teaching you synthetic lethality in very simple words. So two pathways are present in normal cells. Killing one will keep the other path alive. But tumor already have one pathway, this is gone. So if you hit this pathway, tumor cells will die, normal cells will not die. That is synthetic lethality. Okay, so, so let's look at, I will come to synthetic lethality. There is a whole lecture on synthetic lethality. So if you identify what is wrong with cancer cells, so there is a very famous example, and I will tell you about this in details later on, but I want to share just a little bit of more about synthetic lethality. So remember I told you about BRCA1, BRCA2, right? So BRCA1, BRCA2 are absent in breast cancer and ovarian cancer patients, and it is present in normal like healthy people, right? So what are BRCA1, BRCA2? They are responsible for DNA repair. So when BRCA1 and BRCA2 are absent, then there is another protein called PARP. So PARP takes over BRCA1, BRCA2. So whatever repair BRCA1, BRCA2 does, PARP also does the same, right? So DNA is repaired. So hopefully in all of us here, BRCA1 and BRCA2 are active, PARP is active. So we have two ways of repairing our DNA all the time. Now in Angelina Jolie, PARP was not there, right? It was defective. The only thing here was, BRCA1, BRCA2 were not there, PARP was the only one there. So now the cancer cells are dependent on PARP only for DNA repair. Is it making sense? Now because BRCA1, BRCA2 was not there, they had more DNA damage, so they were more prone to cancer, right? Assuming they developed cancer because DNA repair is inefficient. PARP is there to fix it. Now what happens if you give PARP inhibitor? No DNA repair. So if you give PARP inhibitors to tumors, what will happen? Synthetic lethality. They cannot repair their DNA because you have killed the second pathway also. Normal cells, if you give PARP inhibitors, good. BRCA1, BRCA2 will take over. So normal cells will survive, but the DNA, the cancer which lacks BRCA1, BRCA2, they will die because you have killed. That is synthetic lethality. Okay, that's a very simple way of explaining it. Go ahead.
DNA mutation aren't helpful in cancer cells. Like if the DNA is mutating, damaged, and
Very good question. Yeah, very good question. So what happens is that when you mutate the DNA, right? So let's say you have billions of cells and they are all mutating in different ways. So some mutations will kill the cells because those mutations will make non-functional or this or that. But out of hundreds and hundreds and hundreds and hundreds of different mutations, there will be some mutation which will make the protein more active, more oncogenic. Excuse me. So all other cells will die which had wrong DNA mutation. But the right DNA mutation, which makes the cell more proliferative, will survive, right? So what cancer cells do, they make all sorts of mutations because of DNA damage. And those which die, they die. But the one which survives now becomes almost immortal because now they have turned on something which is not affected by other things. So anytime we give a drug and they go mutation, so they don't know what mutations to make. So they make many different kinds of mutation due to genomic instability. So they are randomly making mutations. Many of them dies, 99% die, but 1% survive. That is the resistant guy, right? So when DNA damage occurs, there is always death around. There is always death around. Tons of cells will die, but then the few survivors will emerge, right? So that is why cancer can survive, because it undergoes mutations. So you are right, not every mutation. If I have to guess, huge percentage of mutations are lethal, but the few survivors, they are the one which makes up a whole new colony of survivors, right? Okay, let's talk about tumor microenvironment. So this is a nice picture of tumor microenvironment. These are blood vessels. And see how many different kinds of cells are there which are making this tumor environment here. Okay, so this tumor microenvironment is made of many different cells, red blood cells, ECMs. This is the extracellular matrix which provides nutrients and all these kind of things. These are different kinds of cells which help tumors. So immune cells come. There is a kind of immune cell called M1. So M1 is supposed to kill the tumor cells. They secrete factors and change M1 to M2. Now they are helping the tumor to survive. So all kinds of things they become, and this is the blood vessel which is feeding that, providing food and everything. Okay, it is like you go and live in a new location where there is no plumbing, no supply of food, and then you make the roads, you do all the things, make it a very conducive environment to survive, and then they spread out. Okay. So now you can actually know what are the players of the tumor microenvironment, and then you can actually develop therapy to target this tumor microenvironment. And so you can also develop drugs which is actually targeting the tumor microenvironment. Okay. Yeah, go ahead.
You are saying the tumor microenvironment, we are targeting the tumor microenvironment. But this same microenvironment may be present in other side of body and other parts, so that will be targeted also.
So tumor microenvironment is more unique to the tumor. That is why it is called tumor microenvironment, right? So this kind of environment is not present in other parts of the body. Some of the cells may be there, but that environment is very unique to the tumor. So a person who does not have a tumor, or the body which does not have the tumor, that part, they don't have that same environment. So tumors have a different microenvironment, that is for sure. So what you are saying, the constituents which form the tumor microenvironment, they may be present in other parts of the body too, and they are definitely present. But if you target this one, they are affected more than those guys in different places there. Is it making sense? Yeah, so some of them are there. That is why most of this therapy have some side effect. But then you have to kill the greater evil for that less side effect. Okay? So that is tumor microenvironment. And so see angiogenesis, killing of blood vessels. This angiogenesis is a normal process in rest of the body. So when you give angiogenesis inhibitors, of course the rest of the body will be affected, but it is not that dependent on angiogenesis as the tumor. So people still survive, right? I am in touch with a patient who is undergoing immunotherapy and angiogenesis inhibitor therapy right now. And I talk to that person pretty regularly, and he's feeling better day by day. So just killing the cancer, side effect is less compared to the cancer-related pain and everything else. Okay, and finally the epigenetic modulators. So I have already told you about epigenetics. So you can actually give inhibitors to control epigenetic modifications. Okay, so this is like, so once again epigenetic could be DNA methylation, histone modification, chromatin remodeling. I'll tell you about chromatin remodeling a little bit later. So you can actually give inhibitors, so you can give methylation inhibitors, okay, or you can control acetylation by giving those kind of inhibitors. So now I'll tell you about the Achilles heel concept. So what that means is that cancer cells, in order to survive, they have new mechanisms which help them survive under adverse condition. But when they develop the new mechanisms, they also develop new vulnerabilities, right? So if you can identify this new vulnerability, that is the Achilles heel. So a person is very strong, okay? This is a Greek, I think it is a Greek mythology. So this person is very strong, but the only weakness he has in this heel here, and if you can identify that vulnerability of cancer cells, you attack exactly at that weakness, and then you can kill the cancer cells. Okay, so for example, this brings us to synthetic lethality. If you target A or B, normal cells will survive, but cancer cells only depend on one target, and if you target that one thing, because they don't have the support of both targets, if you target that one, then cancer cells will die. That is the Achilles heel here. This is another very good example. So cancer cells, right, because in order to proliferate rapidly, they have huge amount of polyamines in that, because polyamines are used for constructing the whole cell, right? And normal cells have low polyamine contents, so just simple polyamines, right? And these are used as the building blocks to make other macromolecules. So now we know cancer cells have high amount of polyethylene. So there is this BSO enzyme, BSAO. What it does, it takes the amines and it changes it to aldehyde. And if you make too much aldehyde, what does aldehyde do? Aldehyde is very toxic, and it cross-links a lot of things, react. Aldehyde group is very reactive, right? It takes an amine group and it will form Schiff base with that, right? So you treat it with BSAO. Normal cells don't have too much amine, they will not form aldehyde. But cancer cells have too much amine, they will form too much aldehyde, cells will die. So now you are targeting the vulnerability in cancer cells, right? So if you identify the weakness, so they need for survival huge amount of amines, and they make it. Now we attack those things, right? We have identified the modification there. Okay, so that is what. So what are the emerging frontiers there? We keep on monitoring the patient regularly. We take regular biopsy to see how they are responding, what mutations, what things, and we accordingly keep on changing the drug treatment. You adjust the dose, you see what time, if you start the drug, how soon it is responding, should we add something, should we remove something, so all of these things. AI can, so I was working on something, and then somebody told me that, yeah, this is very effective, but this is very sad story that they are going to develop resistance towards your therapy too. So if you can proactively think about that, if I inhibit this, what kind of resistance mechanism can emerge, then you can proactively design that route. And AI can do easily, and actually you can do without AI in the lab also, and I can tell you the ways to do it. So that will very help, and then you can combine many different things, even in immunotherapy, to do that. So resistance is inevitable, that we know, but we can definitely beat it, no doubt about it. Okay, and the Achilles heel lies in the new weaknesses of cancer cells. They think they are very smart, but they also have new weaknesses, and if we can identify, we can attack that. Okay, and that will result in long lasting that will extend our life, and we'll have better quality of life, and we can beat it in the end.